Hello everybody. This video is a review of all the material we've covered in the mitosis and meiosis unit. And in order to do this, what we want to start with is putting both processes into a biological context. And so cellular reproduction itself is fundamental to the life process because in order for lineages to survive, they must reproduce. And so in unicellular organisms, cellular reproduction is reproduction itself, right? It is population growth for that organism. In multicellular organisms, cellular reproduction is growth and development. And most multicellular organisms reproduce by sexual reproduction, or many of them do, which is where meiosis becomes important. So let's take a look at all of this. Reproducing cells are engaged in a cycle of activity in which the culmination is the reproduction of two cells from one. And we call this cycle, not surprisingly, the cell cycle. And we can think of the cell cycle as composed of two aspects. An interphase, where not much can be seen to be happening, and then a mitotic phase in which the DNA is separated and the two new daughter cells form. The most logistically difficult aspect of this entire process is the separation of the DNA so that each daughter set gets a complete set of chromosomes. In other words, a full set of the genetic information. And that process of dividing the DNA is called mitosis. So if we go back and look at the cell cycle, we can think of interphase as prep time, the time of getting ready to undergo the separation of the DNA and then the separation of the two new daughter cells. And then the mitotic phase as the phase in which the DNA is separated and the daughter cells are formed. And the separation of the two daughter cells are referred to as cytokinesis. The interphase can be thought of as three separate parts, which make a lot of sense when you think about what has to happen in order for cells to duplicate. First, after the two new cells are formed, they're smaller than the original cell, so they must grow, and that requires the reconstruction of membrane and new organelles to be built, etc., the acquisition of food. Then, in preparation for cell division itself, there must be the creation of new DNA. That's DNA replication carried out by the enzymes that you're all familiar with, helicase, primase, the polymerases, and ligase. And then, finally, in the final phase, the cell must be prepared for cell division by replicating the incipient spindle apparatus and by checking for mutations in the DNA. So there's a lot of preparation. And probably also shutting down a lot of the life processes, especially transcription and translation, so that the DNA can be separated. Don't forget that the goal of mitosis is to create genetically identical daughter cells, and these cells are genetically identical except for random mutations, which are a rare event but do occur. It's also important to remember that the word mitosis is used in three different ways. One, simply as the separation of the DNA. During the second definition, which is the mitotic phase, which includes both cytokinesis, cell separation, and mitosis. And then finally, you'll often hear people use it to refer to the entire process of cell division, the entire cycle in a sense. So you'll have to learn what sense is being used by context and by experience in biology and talking to people about biology. Now, in multicellular organisms, there are times when when the organism stops growing, and therefore when their cells stop growing, and therefore there are times when mitosis ceases in certain cells. And that's usually upon reaching adulthood, sexual maturity. And so we can identify cells that are no longer growing go into a phase we call G0. G0 is a quiescent stage in which cells just live their life, but they're not in the active process of dividing. It makes most sense that cells would go into G0 phase at the end of G1 after they have grown back to normal size, but before they've replicated their DNA. So this is where we picture on the cell cycle, cells going into G0. Now some cells, as indicated by this diagram, have the ability to go back into the cell cycle when it's necessary. Other cells, such as nerve cells, don't really have that ability. Let's now take a look at chromosomes before we go any further so that you really understand what's going on in chromosomes. Chromosomes occur on the DNA. Chromosomes are composed of DNA as well as proteins. We call them a complex of the two types of molecules. So they are long linear molecules of DNA with their associated proteins. They carry the genetic information in what are called genes, which are certain sequences of the letters A, G, C, and T along here that can be read by the transcription machinery and then eventually turned into RNAs or into proteins. And we can think of certain locations along the along the chromosome as containing the gene. And you should note that the genes, it's the same gene, but it may be a different form of the gene. We call these two chromosomes homologous because they contain the same gene. And their ancestry history is that one of them comes from the organism's mother and one comes from the organism's father. When we look at chromosome pictures, we will see that they sometimes look single or they sometimes look X-shaped, I-shaped or X-shaped. When you see them in I-shaped, that is their normal shape in the cell when they are carrying out life. When you see them in X-shape, that is their replicated shape. In other words, this is after replication has occurred. The two halves are called sister chromatids, and they should be identical except for mutations. And this is before they have been pulled apart by the mitotic process. Organisms that have two sets of chromosomes are called diploid. Organisms that have only one set of chromosomes are called haploid. Meiosis itself consists of a continuous process that occurs in 5 stages. And you should just recognize that we label these phases in order to talk about mitosis. And so we label them based on certain definitions of things we can observe. The first is prophase, the second phase is pro-metaphase, often referred to as late prophase. Then we have metaphase, anaphase, and telophase. Cytokinesis, the separation of the two daughter cells completely from one another, is typically underway by telophase, and especially by late telophase. In other words, it's concurrent with the end of mitosis. Now, if we want to take a quick look at what these phases look like, you can see it in this diagram, and I'll leave that for you to review, but basically it's the condensation of chromosomes, the lining up of the chromosomes at the metaphase plate, the separation of the sister chromatids, and the reformation of the nuclei, and then the separation of the cells. Let's look at each phase. A cell in interphase shows no discrete chromosomes, but we can tell prophase by the fact that the chromosomes are starting to condense. The nuclear membrane is still present, but the nucleolus has usually disappeared, and the spindle apparatus, if you can see it, has replicated and is starting to move to each pole. In pro-metaphase, you have the loss of the nuclear membrane. You can see the full extent of each chromosomes, and by the end of pro-metaphase, the spindle apparati are now at opposite poles. In metaphase, what we see is that all the chromosomes have lined up along the equatorial plane, which we call the metaphase plate. And then in anaphase, the sister chromatids are separated and are walked by motor proteins towards the pole. In telophase, the new nuclear membrane forms. The chromosomes decondense. You can no longer see individual chromosomes in this photograph. And cytokinesis, here you can see the pinching of the cell, is occurring in telophase. Once again, in the PowerPoint, I have lots of ways for you to study this. The definitions are on each slide, and you can go over that on your own time. In a micrograph, metaphase looks a little messy, but you can tell that the centromeres, the center of the replicated chromosomes, are lined up in a straight line, or if you could see three-dimensionally, in the metaphase plane, in the equatorial plane, or the metaphase plate. Now, the spindle apparatus attaches to the centromere, and then the centromeres are broken apart, and then a motor protein walks them away, as we said earlier. In cytokinesis, a cytoskeleton element literally tightens around the cell slowly so that it doesn't rupture the cell membrane, and you can see slowly the cleavage furrow forms and the two cells are separated. When the cleavage furrow gets down to its smallest bit, the membranes spontaneously form and close so the cell doesn't leak. In contrast, in plants, there's no worry about leakage since the new cells are entirely in an aqueous environment. And so what you have is you have the formation of a cell plate and the cell membrane along with it. And eventually the cell plate is complete, the cell membranes complete themselves, and you get new cells that way. You have all seen the stages of mitosis in a plant cell, and so you should be able to recognize them as well. And here's a slide by which you can practice your recognition. Okay, now that's it for mitosis. Let's go on and look at meiosis. Before we look at meiosis, we have to remember what sexual reproduction is. Sexual reproduction is a new form of reproduction compared to mitotic reproduction, in which half of the genome of two parents is combined into a new individual that is genetically distinct from the parent. And later on, we're going to ask, why would you want to have a genetically distinct organism? In order to do this, you have to create gametes, also known as sperm and eggs, or sex cells, that are haploid, which means they have only one set of chromosomes, not the normal two. And when sperm and egg meet, therefore, and form a zygote, you get diploid offspring, because they get one set of genes, of chromosomes from the sperm and one set from the egg. The process by which haploid gametes are created is called meiosis. So, let's go and talk about meiosis. Meiosis is an adaptation of mitosis, so it uses the same ideas as mitosis, but in a new way. And this is a general theme of evolution. It doesn't invent things from scratch. It tinkers with what's already exists. So mitosis already exists. Sexual reproduction evolves from it. Meiosis, like mitosis, is preceded by a round of DNA replication. Here is unreplicated DNA. Here is X-shaped replicated DNA. As a result, since it starts with replicated DNA, meiosis has to undergo 2 rounds of cell division to get to unreplicated DNA with haploid cells. In the first round, called meiosis 1, the number of chromosomes is reduced from the diploid number to the haploid number, in this case from 2 to 1. And then in meiosis 2, the replicated chromosomes are divided so that the haploid cells now contain unreplicated chromosomes. We thus often refer to meiosis as reduction division, because in the first round we reduce chromosome number, going from diploidy to haploidy, and in the second we divide the chromosomes to make sure we have unreplicated chromosomes in the final product. The stages of meiosis have the same names as the stages of mitosis. Since there are 2 rounds, you might wonder whether there is an interphase between round 1 and round 2. The answer is no, and therefore there is no DNA replication between round 1 and round 2, which would thwart the process of making a gamete with unreplicated DNA. The unique aspects of meiosis compared to mitosis all occur in meiosis 1, and I will highlight them as I go through the stages of meiosis. In prophase 1, the homologous chromosomes pair up and align with each other gene by gene, and then exchange parts in a process called crossing over. And later on, you can see that the chromosomes are a mixture of blue and red, showing that the chromosomes have exchanged their parts. When they look like this, they're called a tetrad, and this process of lining up is called synapsis. So that's the first unique aspect. In metaphase 1, metaphase, remember, is where they line up along the metaphase plate. Instead of all the chromosomes lining up in the metaphase plate, the pairs line up, the tetrads line up, with one of the pairs facing one pole and the other facing the other pole. That's the second unique aspect of metaphase. In anaphase of meiosis, in anaphase 1, the homologous chromosomes, which are still replicated, are pulled apart. And that is the third unique aspect of meiosis, so the third major difference between meiosis and mitosis. In telophase, the nuclear membrane begins to reform. But note, these cells are now haploid. They only have 3 total chromosomes. Usually the nuclear membrane will only partially reform and then will redissolve as we go into prophase 1. You can see that as a vestigial trait of mitosis, the fact that the membrane reforms and then goes away again is a vestigial trait. What has to happen is that the spindle apparatus has to get replicated, and then it has to migrate to the poles. And now what happens in prophase 2 is that migration begins. In metaphase, all the chromosomes line up on the metaphase plate. This is metaphase 2. In metaphase 2, they all line up on the metaphase plate, or the equatorial plane, and then in anaphase 2, the sister chromatids are pulled apart. And then telophase 2, now the nuclear membrane reforms, the chromosomes decondense, and you have the completion of cytokinesis. And so you end up with the 4 gametes of meiosis. The 4 cells of meiosis, because of 2 rounds of cell division. Here's a slide that goes through the unique aspects of meiosis compared to mitosis, but they are, just to repeat, crossing over in metaphase 1, crossing over in prophase 1, lining up of the homologs in metaphase 1, separation of the homologs in anaphase 1. There they are. Now, why has sexual reproduction evolved? And it seems to be that sexual reproduction is a bet hedging strategy to increase variation in offspring, so that some of the offspring will survive as the environment changes. And one of the things we've noticed so far about sexual reproduction is that the offspring are genetically distinct from the parents. But due to 3 processes, independent assortment, crossing over, and random fertilization. The offspring are very variable compared to their parents. New combinations of alleles are formed in recombination that occurs due to crossing over, and new combinations of chromosomes occur due to independent assortment at Anaphase 1, where which pair lines up on which side of the metaphase plate is random. And then sexual reproduction, where there is no favored combination of sperm with eggs, they just randomly meet. Those all increase variation. And so that variation seems to result in organisms that can more quickly track changes in the environment. And therefore, lineages that are sexual survive longer in evolutionary time, and therefore have more chance of leaving descendants than lineages that are asexual, which tend to die out more quickly when environments change. Finally, we can think of organisms that are multicellular as having a life history composed of sequences of mitosis followed by meiosis and sexual reproduction, followed by mitosis, and then the attainment of sexual maturity and the creation of the process of meiosis occurring, creating a new generation. So we can think of life history as the generational sequence of mitotic and meiotic events. So I hope that's been an informative video. Please put comments in the comment section or send them to me, and I will improve this video in time. I'll see you all real soon.